This is slide of submandibular gland. Major salivary glands are parotid, submandibular and sublingual. These are tubuloacinar glands, each of which is surrounded by a capsule of dense irregular connective tissue. From the capsule, connective tissue septa extend into the gland and divide it into lobes and lobules. The parenchyma of major salivary glands is composed of secretory acini, but because some portion of ducts are also involved in the secretory process, these glands are included in the tubuloacinar variety. The acini are spherical or tubular structure, each consisting of a secretory epithelial cells arranged around a central cavity. The submandibular gland is a compound tubuloacinar gland of mixed variety. However, its secretion is predominantly serous in nature, and 90% of its parenchyma consists of serous acini. Remaining 10% of parenchyma consists of mucous acini. Some of the mucous acini have caps of serous cells called serous demilunes that are crescent shaped. The cells of serous acini are pyramidal in shape with a broad base facing the basal lamina. Each cell contains a spherical nucleus located in the basal part of the cell. The apical region contains secretory granules called zymogen granules. The apical portion of serous cells stains acidophilic due to presence of large number of zymogen granules. The cells of mucous acini are also pyramidal in shape. Their nuclei are oval or flattened and pressed against the basement membrane. The apical region of cells is occupied by mucin granules which are washed out during staining procedure. Under light microscope, the apical region of mucous cells stains poorly and appears to be empty. The mucous cells secrete mucin, which is strongly hydrophilic glycoprotein. Upon contact with water, the mucin becomes converted into mucus, which is gel-like lubricant. The duct of salivary glands are classified into following three types: intercalated ducts, striated ducts, and excretory ducts. Acini open into intercalated ducts that have a smaller diameter and are lined by simple cuboidal epithelium. Several intercalated ducts open into a striated duct, which is lined by low columnar cells. The striated duct from each gland lobule drain into interlobular duct. Interlobular ducts are lined by simple columnar epithelium. Interlobar ducts are lined by pseudostratified columnar epithelium. The main duct is lined by stratified columnar epithelium that changes into stratified squamous non-keratinized near its opening.